First, let us start it with the pharmacogenomics. It can be defined as is the branch of genetics that is concerned with determining the likely response of an individual to the therapeutic drugs. So, what is this pharmacogenomics? This is nothing but it is a branch of genetics that is dealing with how the individual responds to that of the therapeutic drug. And this concept of pharmacogenomics can also be used in identification of the new targets. And the next one is the pharmacogenetics. It is a branch of pharmacology that is concerned with the effect of the genetic factors on reactions to the drugs. So, what is this pharmacogenetics? It is also a branch of pharmacology reacting to that is affects the genetic factors in reaction to the drugs. So you can see there like there is a almost similarities between this pharmacogenomics and the pharmacogenetics. What is the difference between this pharmacogenetics and pharmacogenomics means the pharmacogenetics usually refers to how variation in one single gene influences the response to a single drug. And this pharmacogenomics means it is a broader term which studies how all of the genes, mainly the genome, can influence responses to the drugs. So, what is this pharmacogenetics and pharmacogenomics? The genetics is the only there is a difference between single gene, and the pharmacogenomics means the group of genes influencing the response of the drug. That is the difference. And coming to the epigenetics, this epigenetics is the study of heritable phenotype changes that do not involve alterations in the DNA sequence. Like, what is this epigenomics means? For example, we have a chromosomes in our body. If we take the inside parts of the chromosome, we get chromatin. From the chromosome, we get the chromatin. From the chromatin, we have the DNA. In the DNA, which is being composed of the genes along with the histone proteins, which is being wrapped around the genes, right? And before this histone proteins, a tail is being present. For this tail, if a epigenic factor is being attached, then that is called as the epigenomics. So if you see here, there is only a change in that of the histones, but not in the DNA, but not in the gene, that is the DNA. So we can tell like there is a heritable phenotypic change, but there is no a genotype change in this. That is, there is no alterations in that of the DNA sequence. And We can tell like there is one example for this epigenetic change in the eukaryotic biology is the process of cellular differentiation as a single fertilized egg cell called the zygote continues to divide. The resulting daughter cells that change into all the different cell types in an organism, including neurons, muscle cells, epithelium, endothelium of the blood vessels. And these are formed by activating some genes while inhibiting the expression of others. So, what is this epigenomics? We have discussed like there is only a change in that of the alteration in the DNA in the phenotype, but not in the genotype. So, in general process also this epigenomic genetics occurs where means in the after the formation of the zygote, which they'll be divided into the daughter cells, and it will from the zygote we develop neurons, the muscle cells, epithelium, the blood vessels, everything. And how are this forming from the one particular zygote means by inhibiting the expressions of the some genes and activating some genes, all these are being formed. So by this we can tell like this epigenomic factor binding to that of the histone has an effect on the gene, but there is no alterations in the gene by itself. Okay. And coming to the next one is polymorphism. This polymorphism is the presence of genetic variation within a population or the condition of occurring in several different forms. So, what is this polymorphism means? The occurrence of a single gene in many forms. Okay, and in order to understand this, let us discuss it with an example. A gene is said to be polymorphic if more than one allele occupies that gene's locus within a population. For example, in dogs, the E locus can have any of five different alleles, known as The em, e to the power of m is e to the power of a g, e to the power of h, and a small e. These varying combinations of these alleles contribute to the pigmentation and patterns seen in dog coats. Right? So, what is this polymorphism? By this example, we can know that, for example, if there is a dog in a dog population, the E. coli gene, in that there are different alleles. This is called as the polymorphism. Because of this, there is different patterns of pigmentation in the dogs, so that some dogs are white in color and some dogs are uh, brownish in color. Okay, and you can tell like the presence of genetic variation within a population is called as polymorphism. And regarding this examples, there are many examples for this, like slow accelerators and the fast metabolizers, rapid metabolizers. We'll be discussing about that in detail in future. And next, coming to, as you have given in your first topic, like a point, like the subfamily nomenclature. With respect to the nomenclature, family is a number, subfamily is a letter, isoform is a number. So now let us explain this. What is this? For example, we have SLC NXM. This is, let us suppose this is the, for example, this is a SIP cytochrome P450 D61 proteins. So we have many proteins which are many enzymes for the metabolism. So let us suppose, we are taking one example with some alternative SLC like that, where this SLC is the root name. For example, the SIP, the cytochrome P450 can also be, and N. The N indicates, a small n indicates an integer that is representing a family. Like example 1, 2, 3, and so on. And coming to the capital X, a single letter, capital X indicates a letter, that is like A, B, C, and so on, denoting a subfamily. And the small m indicates an integer representing an individual family member. This is an isoform. So for this example, the S SLC1A1 is the first isoform of the subfamily A of SLC family 1. So by this we have done with the nomenclature, like the family is the number. So this n here, the family is a number obviously. The subfamily is a letter. The subfamily is a letter. And the isoform is a number. Here the isoform is an integer, like numbers. Okay. And the one more thing you have given to me is in that, like, this is the one more topic you have given me, like NAT2 polymorphism divides population into rapid oscillators and the slow oscillators. The slow oscillators is equals to the higher plasma concentration of the dapsone, hydralazine, isoniazid, and more adverse drug reactions, and fast oscillators more susceptible for to procainamide toxicity due to the buildup of NAPA, that is then N-acyl procainamide, which is an active metabolite, and the isoniazid mutation in that of the Na2 lead to polyneuritis, which is a study done by the Evans and the Clark. Let us explain about this in detail. In the central nervous system, the central nervous system tuberculosis is a serious and often a fatal form of tuberculosis that predominantly affecting young children. Why are we discussing about the tuberculosis? Because as you have given me like the isoniazid can be used in the treatment of this tuberculosis. Okay. This isoniazid is one of the most effective antimicrobacterial drug. This acetylation reactions in the metabolism of isoniazid are being catalyzed by N-acetyl transferase 2. I can write it like NAT2, which shows genetic polymorphism resulting in two distinct phenotypes, that is the slow and rapid acetylators. Until date, this NAT2 loci have been shown to express 36 alleles resulting from the existence of numerous single nucleotide polymorphisms with 7 missense and 4 silent substitutions that have been identified in the NAT2 coding exon. The absence of any of these substitutions is considered as the wild type allele, and they are also called as the rapid acetylators. So what is this? Why are we discussing about this? This isoniazid, which is an antimicrobacterial drug, which is being metabolized by this NAT2. And this NAT2 has some genetic polymorphisms, like it can be expressed as the slow accelerators or rapid accelerators. When it is called as a slow and rapid means this the NAT2 loci has 36 alleles and some 7 missense codons and the 4 silent substitutions in the NAT2 codon exon. In the absence of any of this, leading to rapid accelerators. Okay. So what happens in case of slow accelerators? If this is the drug which is given normal dose to the patient, as a patient is a slow accelerator, because of this, there is a decrease in the metabolism. So the drug concentration increases in the body. As we can return it like the high plasma concentration of isoniazid. If the concentration has been increased, then there is an increase in that of the maximum safety concentration, which leads to occurrence of the side effects. The same concept applies for the dapsone and the hydralazine as well. Okay, and coming to this, so let us discuss this isoniazid topic given completed here. And because of this mutations in this gene, because of this isoniazid accumulation, this is leading to the polyneuritis. This is mainly seen in case of the slow accelerators only. Only in them there is a accumulation of this isoniazid is being seen. And the next one is the fast accelerators, which is mainly seen, for example, the procainamide. So what is this procainamide? This procainamide is a class 1A anti-arrhythmic agent whose primary mechanism is related to its antagonism of cardiac sodium channels, delaying the phase zero of the cardiac cycle. This procainamide and its metabolite N-acetyl procainamide also exert the potassium efflux channel, blocking effects leading to the prolonging the QT interval. So what is this procainamide? This procainamide is nothing but it is a class 1A antiarrhythmic agent. What is its role? Its role is to antagonize its sodium channels and thereby decreasing the arrhythmias by increasing the QT interval prolongation. This procainamide undergoes metabolism in the liver and it is being converted into a metabolite called N-acetyl procainamide. Because if a drug is being under this drug also has effect on the QT interval. So if this drug in the normal doses, if it is given to a fast acylator, then what happens? Then this procainamide is being rapidly converted into the N-acetyl procainamide. Then because of this N-acetylacetamide has been the concentration of it has been increased and there is an adverse effect seen in this patient because of the fast acetylation. And there is also because what is adverse effect seen because of this N-acetyl procainamide concentrations, or if this N-acetyl procainamide concentrations are greater than 20 microgram per ml, then the person is at a higher risk of torsades de pointus. What is this torsades de pointus means? It is a specific form of polymorphic ventricular tachycardia in the patients with a long QT interval, and it is characterized by rapid irregular QRS complexes, and this is adverse effect seen. And we can also discuss one more concept here, like the drug-drug interactions happening. First, what is this drug-drug interactions means? The drug interaction that can occur when drug interaction. This is also one of the concept which you have given to me. This drug interaction has occurred when the administration of one drug alters the clinical effects of another drug. So let us take the same isoniazid example. This isoniazid is also a microsomal enzyme inhibitor, and it inhibits the metabolism of co-administered drugs such as acetaminophen, carbamazepine, diazepam, phenytoin, theophylline, and warfarin, resulting in their high plasma concentration. As we discussed in case of the tuberculosis, that is the tuberculosis affecting the brain, the cerebral tuberculosis, central nervous system tuberculosis. And the patient may be represented with a symptom like seizures, right? Because of this seizures, the patient may be prescribed like the isoniazid along with the phenytoin. When these two are simultaneously given, administered, then if this patient is a slow acetylator, possibility of phenytoin intoxication, that is becomes high. So what is this drug interaction in relationship with this isoniazid means? This isoniazid prevents the metabolism of all these drugs by blocking its enzymes. So if this drug and it's given in with these combination drugs, obviously as it causes an prevention of this enzyme inhibition, these drugs metabolism will be decreased and their drug concentration will be increased in the blood. So what if the patient is a slow oscillator? Then it worsens the condition. If the patient is a slow oscillator and if the patient is being given both this isoniazid along with this phenytoin, then there is a higher chance of this phenytoin toxicity, as the higher chance of phenytoin toxicity is being seen. Okay. And we have many other drugs and their mutations of these enzymes. So let us discuss about all those in detail. This is also which you have given, like you have given the drug, the polymorphism and the effects of the drugs, and I'm gonna explain all that, the antidepressant, all the drugs, the polymorphisms, and how it is being affected. Okay. Coming to the first thing is the antidepressants, the CYP2D6 mutation is causing nervousness, agitation, and lethargy. What is this? These antidepressants are being generally metabolized by the CYP2D6 enzyme. As there is a mutation of the CYP2D6 enzyme, then the antidepressant levels will be increased in the body, leading to nervousness, agitation, and the lethargy to the patients. And the next is the tamoxifen. This tamoxifen is a non-steroidal anti-estrogen. The polymorphism involved is the CYP2D6. This 2D6 converts tamoxifen, which is a prodrug, to endoxifen, which is an active form. And so, what is this tamoxifen? This tamoxifen is a non steroidal anti estrogen, which is generally used in the treatment of the breast cancer. And this tamoxifen is not an active drug. This is a prodrug. After its metabolism by the CYP2D6 enzyme, it is being converted into the active form. As if there is a mutation to that of the CYP2D6, then it can't convert this tamoxifen into the endoxifen, which is the active form, leading to lower drug efficacy, and so the breast cancer can't be treated properly, right? Because of the mutation in CYP2D6, it can't make endoxifen, and thus there is a lower drug efficacy, leading to the breast cancer. Sorry, leading to decrease in recovery from the breast cancer. So I have seen some of your multiple choice questions which have been asked in your previous exams. Like they may also ask like this what is this CYP2D6 enzyme doing to the tamoxifen, and what is its role in the breast cancer. So you can select it like a decrease in the recovery from the breast cancer. And all this in-depth knowledge will be useful for you in writing your choose the correct answers. And the next one is the tramadol. This tramadol is a analgesic that is being used to treat moderate to severe pains. And this is being even this is being metabolized by the CYP2D6 enzyme. Let us discuss in detail about this CYP2D6 enzyme once. The CYP2D6 enzyme metabolizes a quarter of the prescribed drugs, like as we discussed, even the tamoxifen and when the tramadol is being metabolizes the tramadol. The individuals who carry two inactive copies of this CYP2D6, they are known as poor metabolizers. And as we know, because of this poor metabolizers, there is an high levels of tramadol in the blood. But even though there is high levels of tramadol in the blood, there is a lack of efficacy is seen. This is the reason because this tramadol is actually a prodrug and it should be converted into the active form. Okay. And as because of this mutation, there is no conversion into the active form. There is a no active form being seen, and leading to the lack of efficacy. And there are also some other metabolizers like intermediate metabolizers and ultra rapid metabolizers. They are, if the individuals who carry one or more reduced or inactive copies of CYP2D6, they are known as intermediate metabolizers. And those who carry more than two active copies of CYP2D6, they are known as ultra rapid metabolizers. So we can tell, like, even in this ultra rapid metabolizers, there is a fast elimination of this tramadol. So even in that condition, we can see a lack of efficacy, right? In case of ultra rapid metabolizers, there is a reduction of the tramadol concentration in the blood, leading to lack of efficacy. And the next one is niperidine and codeine. And all these two are also the opioid agonist that are being given in reduction of the pain. Even this are being metabolized by that of the CYP2D6 enzyme. Even the same we can and the applicables for this as well. Even this meperidine and the codeine, they are not the active forms, they are being converted by this enzyme into the active forms. So in case of poor metabolizers, their levels will be high in the blood, leading to the lack of efficacy. And one more thing in them is this cytochrome P450 2D6 is an enzyme that is expressed not only in the liver but also in the brain, and it can inactivate neurotoxins such as 1 methyl 4-phenyl 1 2 3 6 tetrahydropyridine and 1 2 3 4 tetrahydroisoquinoline. So genetically, the slow CYP2D6 metabolizers are at higher risk of developing hyperreflexia or the seizures. So, how is this effect causing the hyperreflexia or seizures means this CYP450 2D6 enzyme is present not only in the liver but also in the brain. It inactivates some of the neurotoxins called as 1-methyl 4-phenyl 1236 tetrahydropyridine, and this is the major neurotoxin which damages the brain. And because of this, as there is a damaged, or because there is a slow acetylation for this, there is a decrease in the inactivation of this neurotoxin, causing increase in the concentration of the neurotoxin in the brain, leading to the seizures or the hyperreflexia. And the next one is the warfarin. The warfarin, like the CYP2C9, CYP2C9 star 2, CYP2C9 star 3, and VKORC1. In that VKORC1 A slash A. In this, the star 2 and star 3 variants, they have the lower warfarin dose. So, what is this warfarin, and this warfarin is being metabolized by these enzymes? In case of a mutation in this, then it won't work properly, or in case of a slow acetylators also, there is, in case of for example, in case of a slow acetylators, their warfarin concentration will be increased. The main ADR for this warfarin concentration, if it is increased in the blood, is bleeding. So, in order to prevent this, if the patient is a slow oscillator, mainly for the variants CYP2C9 star 2 and CYP2C9 star 3 variants, then how can we treat this? We can treat it by giving the low dose of the warfarin. Let us brief it still more in detail. This star 2 variant, there is 11% decreased clearance, and so we should reduce the dose by 17%. So what is this star 2? And if the patient is a slow oscillator with this CYP2C9 star 2, then this reduces 11% of the clearance of the warfarin, and so we should reduce the dose which is being normally given to 17%. Okay, and the star 3, which is a 7% decreased clearance, and reduce the dose by 37%. So this star 3 variants, there is only a 7% decrease in the clearance, but even though there is 7% decrease in the clearance, and we should need to reduce the dose by 37%, because we can see most of the ADRs when performing the studies, most of the ADRs have been seen with that of the star 3 variants when compared to star 2 variants. So we have conducted a study by giving the 37% of the reduction in the dose. For those patients, we have seen the beneficial results like preventing the bleeding. And that is the reason, even though it is a 7% clearance, we need to reduce the dose by 37%. And coming to the next is the VKORC1AA. Even in this helps in the metabolism of this warfarin. Mainly, it is involved in the vitamin K, and because of this, uh, even this is also similar to that of the SIP. So, in this, even this, the warfarin concentration may be increased. So, we need to reduce the concentration of the warfarin, and but in this, we need to reduce the it to 37% of the dose reduction is to be maintained. And coming to the next one is clozapine. What is this clozapine? This clozapine is a serotonin antagonist with strong binding to 5-hydroxytryptamine 2A 2C receptor subtype, right? This 5-hydroxytryptamine, if there is a receptor mutation is seen, then there is a non-response in schizophrenia patients. So, what is this clozapine? This is a serotonin antagonist, which it binds with that of the 5-hydroxytryptamine receptors. If this there is a mutation in these receptors, then this clozapine doesn't bind to it, so it doesn't show any effect over there, causing there is no effect in treating the schizophrenic patients, right? And the next one is the leukotriene inhibitors. In order to understand this, let us discuss yeah, one thing like 5 lipoxygenase. This is being encoded by ALO5. This 5 lipoxygenase plays a key role in the conversion of the arachidonic acid to several potent lipid mediators that is being involved in inflammation, including the pro-inflammatory leukotrienes. And If there is an ALOX5 mutations, then there is a non-response in the asthmatics. So we know that in asthma is not there is a production of the leukotrienes. How are these being produced? They are being produced from that of the arachidonic acid, which is being mediated by the 5 lipoxygenase. Okay, if this lipoxygenase is being encoded by the genes ALOX5, what happens if there is a mutation over there? then then it doesn't produce the proper leukotrienes. So if there is only no proper leukotrienes are being produced, or if there is any um, improper or mismatching leukotrienes are being produced, then this leukotriene inhibitors, they can't bind to these leukotrienes, right? And so when we give this leukotriene inhibitors to that of the ALOX gene mutation patient, then there is a no response is seen in that asthmatic patient, right? And the coming to the next one, the oral contraceptives. What is this oral contraceptives? These are the estrogen containing that increase the plasma concentration of clotting factors. And the factor 5 lead mutation. What is this? This factor 5 lead is a result of a single nucleotide substitution at single nucleotide substitution by the adenine in the place of guanine, and thereby this results in missense mutation. And because of this mutation, there is more production of thrombin and leading to clotting. It is leading to thromboembolism. So what is this oral contraceptives? And factor 5 lecithin mutation and the thromboembolism. This is the only line you have given to me. And in order to understand this, you need to understand what is this the factor 5 leading mutation. The factor 5 leading mutation is nothing but the mutation of this by that of the replacement of the guanine with that of the adenine sequence. This factor 5, this is an unclotting factor, right? And if there is a mutation in this, then it causes increase in the production of the uh, thrombin, and further leading to that of the clot formation, leading to thromboembolism. One of the main adverse effects seen if there is a factor 5 leading mutation in the oral contraceptives is thromboembolism, mainly the venous thromboembolism. And what is the relationship between this oral contraceptives and the factor 5 is this oral contraceptives, majorly they are composed of the estrogen containing. These estrogens, they increase the concentration of the clotting factors like the factor 5, factor 7, factor 10, and many. And because of this increase in the concentration, there is an increase in that of the thromboembolism formation. And the next one is penicillamine. And the glutathione, yes, transferase M1 non-response in rheumatoid arthritis patients. You have given me this line. So in order to understand this, this glutathione S transaminase is thione S transaminase polymorphisms may influence the response to D-penicillamine in rheumatoid arthritis patients, and that patients in possessing of GSTM1 or the GSTM3 haplotype or significantly less likely to show a beneficial response to the drugs. These penicillamines, they have a response with that of the glutathione as transaminase. Generally, if the patients who are representing with the glutathione S transaminase M1 polymer or the glutathione S transferase M3 polymers, then those patients, or while performing the studies, clinical trials, we have seen that those patients have no response, or they are being less responded to that of the penicillamine while treating this rheumatoid arthritis. And coming to the next one, next drug is tacrin, and non ApoE4 improvements in Alzheimer's disease. You had given me this one. This lipoprotein E4 allele is strongly linked to both sporadic and you can write it like it is a link to the Alzheimer's disease in simple. That raises the possibility that a dysfunction of the lipid transport system could seriously affect this lipid homeostasis in the brain. And this tacrin, it reduces the ApoE4 activity. And what is this? The apolipoprotein E4 allele. This is linked with that of the Alzheimer's disease by um, affecting the homeostasis by dysfunctioning the lipid transport system. Because of this, there is a alteration in that of the lipid homeostasis, and which is causing the Alzheimer's disease. What happens if this drug sorry, this is, when this tacrin is given to the with the polymer with the polymorphism to that of the ApoE4 gene with the ApoE4 allele, that is which is a non-ApoE4 allele patients, then we can see there is an improvement in that of the Alzheimer's disease, where this tacrin acts in combination or in association with that of this apolipoproteins. Or like this taccharin is being influenced by this apolipoproteins, like. And the next one is omeprazole. Here the CYP2C19 or the CYP2C19 star 2. This omeprazole, it is being a rapidly metabolism, 28.6% cure rate for H. pylori, and intermediate, that is a 60% cure rate for H. pylori, and a slow, that is a 100% cure rate for H. pylori. This is the data I have given. This omeprazole is being metabolized by this the CYP2C19 or the subclass CYP2C19 star 2 variant. If this is being metabolized by this enzyme, then in case of rapid metabolizers, its concentration is decreased. As this omeprazole is mainly used in case of the peptic ulcer disease, the peptic ulcer is mainly caused by one of the bacteria called as the H. pylori, and because this H omeprazole is a proton pump inhibitor, because of this H. pylori, there is an increase in the acid production, and this omeprazole tries to block the acid production. And in the rapid metabolizers, the 28.6% cure rate is being seen because the omeprazole is being fast eliminated. And in case of the intermediate metabolizers, 60% cure rate is being seen. And in case of slow metabolizers, the 100% cure rate is being seen. And here we can also discuss one drug interaction, like if this omeprazole is being in given combination with that of the clopidogrel. Clopidogrel plus omeprazole. Then this clopidogrel is a inhibitor of, sorry, this omeprazole, as it's having a more affinity with that of this CYP2C19 enzyme, even as this clopidogrel is also being metabolized with the same enzyme, because there is a competition between this clopidogrel and omeprazole. The competitive drug-drug interaction is being seen, like this omeprazole is being metabolized first, leading to uh, sorry, this omeprazole um, causing a decrease in antiplatelet effect. If the patient has been having with a mainly, especially with this the CYP2C19 star 2 variant, then if the patient is being given in combination with the clopidogrel in combination with that of the omeprazole, then there is a decrease in the antiplatelet effect is being seen. As we can tell, like there is a decrease in the concentration of the clopidogrel. Why this concentration of the clopidogrel is being decreased? Because this omeprazole increases its uh, activity, and it's causing the constant increase in the metabolism of this clopidogrel. And coming to the next one, irinotecan, the polymorphs is IgT1A1, UGT1A1 star 1, UGT1A1 star 17. The risk of irinotecan toxicity increases with, like you may be getting a confusion, like here this clopidogrel concentrations will be increased even though there is a decrease in antiplatelet effect, because this clopidogrel is a pro drug and it should be converted into the active form. Because of the competitive inhibition by that of the omeprazole, there is a decrease in the antiplatelet effect. The risk of irinotecan toxicity increases with genetic variants that is being associated with reduced UGT enzyme activity and the presence of this variant results in reduced excretion of irinotecan metabolites, which is leading to increased active irinotecan metabolites in the blood. And because of this increase in the concentration of this irinotecan, then which is first, what is this irinotecan? This is nothing but a acetylcholinesterase inhibitor, and this mainly this star 1 variant. Because of this accumulation, the patients with the star 1 variant may experience some of the adverse effects like causes the ADRs like neutropenia and the diarrhea, and more than star 17. And some of the more severity of the A seen in case of this star 17 variant. And the next one is the abacavir is HLA B star 5701. What is this HLA means? This HLA is histocompatibility complex class allele. And majority of the patients with this histocompatibility complex allele has experienced severe hypersensitivity reactions. Not only with this drug abacavir, but also there are many other drugs um, in, uh, who are expressing this complex class have experienced. What is this histocompatibility complex means? These are responsible for that of the hypersensitivity reactions by expressing the macrophages by initiating the reaction. This histocompatibility complexes, they think like this abacavir as a foreign, and they react with this and they cause severe hypersensitivity reactions. And there are still many others. Sorry if you have given you.